Thank you for that introduction. Today I'm excited to talk to you all about how you can bust out quick, low-volume protein characterization on Stunner. We'll review how Stunner can take a look at quantification and sizing for protein therapeutics, and we'll take a specific deep dive into ADC characterization, including some new software applications that help you quantify those drugs specifically. Development of biologics reminds me a lot of auto racing. During the majority of the race, drivers are moving around the track as fast as they can to be the first to finish. Several times during the race, the cars have to come in for a pit stop to get a fresh set of tires, top up the fuel, and make any adjustments to give the driver any advantage they can. These pit stops are as efficient as possible, with everything happening perfectly and at the same time. To me, that sounds a lot like a biologics process running as fast and efficiently as it can, but also with the need to perform characterization checks as fast and as thoroughly as possible. The kinds of rapid, thorough biologic check-ins that I'll be discussing today will be about quantification and sizing for protein samples and fit right in as at-line analytics for development of biologics or when running a biologics process. When it comes to answering major questions for characterization of a biologic, there's two that I want to focus on today. The first one, how much do I have, is all about quantification. The second one, what do I have, is kind of about the quality of your sample. Is it the right size, or have aggregates struck, or even is there fragmentation going on? When you're thinking about answering the first question, there are a lot of methods, but a traditional one is UV-Vis, because it is great at getting you fast, easy answers on protein quantification without any sample processing. DLS is a great technique for answering questions about size and aggregation, again because it gives you fast, simple answers without any sample processing required specifically for the assay. So this begs the question, what if both of these analytical techniques were combined? And that allows me to introduce Stunner. Stunner is the only system to pull together the two separate technologies in UV-Vis for quantification to determine protein concentration or other molecule concentrations, and DLS for sizing and aggregation determination. All of these results are collected from a single 2 microliter sample. If you're working with conjugates, Stunner can also measure average drug-antibody ratio, or DAR, to know the ins and outs of that conjugate, usually antibody-drug conjugates or ADCs. Without skipping a beat, Stunner allows you to make a very rapid pit stop to check on the characterization and quality of your sample and know if your sample is good to go to the next step or ready to run in the next assay. All you need to do to run a sample on Stunner is load 2 microliters of each sample into a Stunner plate. A Stunner plate is a normal 96 well plate that has each well replaced with a microfluidic circuit that only needs 2 microliters as an input volume. Each of these circuits has 2 fixed path lengths where the sample will ultimately be pumped into there, and then light will be passed through those microcuvettes to give it a wide dynamic range of UV-Vis absorbance. When that UV-Vis absorbance is measured, you can understand the concentration of the sample that you've loaded. This is also important to know that because of that wide dynamic range, you don't need to dilute your sample to get within the range of the assay. If you're dealing with a full plate of UV-Vis measurements for concentration determination, you can get 96 measurements in about 10 minutes. At the same time, you can read the same samples with DLS or dynamic light scattering to measure the average size and distribution of the sizes of particles in solution. This tells you answers to questions like, is my antibody the correct 10 nanometers in diameter? Or has my antibody aggregated and my sizes are much larger and my size distribution is much wider? If you're running both UV-Vis and DLS on all 96 samples, a full plate read takes about an hour. Here we can see exactly what sample loading looks like with the help of some fluorescein and a black light. In 2 wells, we've already added 2 microliters of fluorescein. And as you can see, to load in another sample, you simply pipette in 2 microliters into that input well, and the sample wicks into the plate. I showed you that in real time to understand that the sample is securely sucked into the microfluidic cuvette, so you don't have to worry about evaporation when dealing with large sample numbers and tiny volumes. After a sample's been put into the plate, you can see the difference between sample loading on the left and samples that have been read in the plate on the right. On the left, all of these samples are nice and secure and waiting to be read. And on the right, you can see how the sample has been pumped into the 2 microcuvettes where the UV-Vis measurements and the DLS measurements have taken place. Not only is Stunner super easy to use and load, but it also provides fantastic data. Here we are showcasing the huge dynamic range of Stunner from 0.03 to 275 OD. This means that your sample can be measured without the need for any dilutions or reagents, and it can fit the dynamic range that you need for your samples. On the left, we can see absorbance spectra with different concentrations of purified IgG with the telltale absorbance peak at 280 nanometers. This is a dilution series where each replicate is being run with an n equals 8, and we're graphing all of those lines here in this graph on the left. You notice that they all overlay each other very precisely. On the right, we're plotting that absorbance at 280 nanometers and against the known concentration and showing that the data is linear throughout the entire dynamic range. Once again, from 0.03 to 275 OD. And once again, we're showing these as 8 individual dots that actually overlay each other very nicely to highlight the precision of this assay. We can dig into that data set a little bit further to continue to quantify Stunner's accuracy and precision. Here on the left, we are now plotting the measured concentration from Stunner against the known concentration. You can see that this result is very linear, the slope is very close to 1, and the R-squared value is also very close to 1, indicating that the expected and measured results correlate very nicely and that the expected measured results are very close to each other. We're quantifying this in the table on the right, where you can see the actual values for known and measured average concentration are within 2% of each other, which is our specification. And the precision, measured as percent CV, or relative standard deviation, RSD, are below 1%. Again, this is with octuplicate samples. This shows us that our 2 fixed path lengths in Stunner's microcuvettes allow us to have incredibly accurate and precise data across a wide dynamic range and covers most applications in the world of biologics at low and high concentrations. And beyond just reading a sample for quantification and concentration based on UV-Vis, Stunner can also measure samples for size based on DLS. So in this case, we're continuing to show quantification information on the left at a very precise value. But on the right, we've also taken the size measurement from DLS and graphed 8 samples in octuplicate in an overlay. Here on the x-axis, we're looking at hydrodynamic diameter, and on the y-axis, we're looking at light scattering intensity. You can see how the 8 samples overlay very nicely, and they're giving us an average size, or a z-average diameter, which is a weighted average of diameter based on light scattering intensity, and that average is about 11 nanometers, which is what you'd expect typically for an antibody sample. The precision of this measurement is also quite excellent at about 1.5% for our 8 individual wells. Here's a look at some example sizing data from DLS to further explore that concept. In this case, we have measurements of 2 commercially available proteins, thyroglobulin and ovalbumin, which are loaded at the same concentration in triplicate in 6 separate wells. Note that these proteins are different in size, which is why when we overlay the data, we can see peaks at different sizes along the x-axis. On the left, we're looking at an intensity distribution, much like the last slide, where we're seeing diameter versus light scattering intensity graphed. In that graph, we can see that thyroglobulin is scattering more light, which makes sense because they're at the same mass concentration, but thyroglobulin is a larger molecule and light scattering scales proportionally to the size of the molecule. So, first lesson here is that larger particles will scatter more light. That'll be a useful principle that we'll explore later when we're considering aggregation in a protein sample. If you look at the right graph, you can see an overlay of mass distributions where we're taking that intensity data and we're converting it to mass data to help us understand the distributions that we get from DLS. On the right, we're graphing diameter versus mass concentration, and you can see in this case, Stunner has calculated that the concentrations of the two proteins are very close to each other, which is great because that's something that we intended with this experiment, and that's something that we confirmed with UV-Vis, which is what I would always use for quantification by UV-Vis. But this helps us understand the magnitude of concentrations at the size measured by DLS. If we want to apply those two principles to an actual real-world use case, we can think about the need to spot aggregation in a protein sample. DLS is one of the most rapid ways we can use to detect aggregation, but it's also one of the most sensitive to larger aggregates. Here we're looking at two samples. In the left column, we have a pure, homogeneous, and I'll say happy protein or antibody sample. You can see how the intensity and mass distributions agree, where this sample averages out to be about 11 nanometers in diameter. That means I have great sample quality and no problems have happened. On the right column, we've taken that same sample and then we've spiked in another sample that has been heated for several hours to cause it to aggregate. With this spike in, we've only used a small volume of aggregates so that there should only be about 1% by volume of aggregates. So in this DLS read, we see the sample looks dramatically different. When we're evaluating sample by intensity, we can see that those rarer populations, because they are larger in diameter, give a much stronger intensity signal than the monomeric antibodies peak. So this is useful because it tells us that DLS is a great technique for detecting rare large aggregates. The mass distribution also helps us understand the magnitude of the problem because it's calculating, or it's converting those intensity peaks into mass peaks to help us understand how much of those aggregates are present. In this case, we can see that our monomeric peak is still the majority of the sample and indeed help us characterize what the most common size of the majority of the sample is, again at about 11 nanometers diameter. And those aggregate peaks are present. You can see they're indeed just a few pixels on this graph, but they are the minority of the mass of the sample. So when thinking about things in terms of milligram per mil concentrations, the mass distribution helps us understand roughly what's the proportion of monomers versus aggregates present in our sample. Okay, so let's take Stunner and show how we can apply it to characterization with an antibody sample. Many use cases for Stunner include using it to find the right formulation buffer for each of your antibody constructs. An optimized formulation buffer will keep your antibodies in their native monodispersed form. Even when temperatures go down for storage or the concentration goes up for therapeutic dosing, the right buffer is needed to keep things functional. When antibodies are in the wrong formulation buffer, this leads to more self-interaction and the formation of aggregates. Aggregates lead to issues including decreased stability of your molecule and rejection for use as a therapeutic, as they may lead to an immunogenic response after administration. There are so many factors that go into finding the perfect formulation buffer, like pH, ionic strength, the right type and concentration of excipient, etc. It is difficult to predict what the ideal formulation buffer will be without direct testing. And when you're doing direct testing on something that has a lot of different variables, you want to make sure you can screen those results using the minimum sample volume possible and get answers in the fastest amount of time possible. That's where Stunner fits in. When you're running formulation screens for monoclonal antibodies, you can use Stunner to identify which ones cause aggregates early on in your process. And indeed, you can also use it to detect rare large aggregates, as I've spoken to earlier. Here we're showing a formulation screen of a monoclonal antibody at about the same concentration, 50 milligram per mil, in two formulation buffers, a commercial one and an acidic citrate phosphate buffer. You can see right away from the DLS intensity distributions that the protein is monodispersed in the commercial buffer shown on the left and in the top row of the table, but aggregates once it's been buffer exchanged into the acidic citrate phosphate buffer. We can do a little bit of quantification of that as well. You can see that in the peak 1 mean diameter and the peak 2 mean diameter, where we can understand the average size of those two peaks. And indeed, if we wanted to look at the mass distribution for these results to understand how many aggregates are present roughly, we could. Also in the table, you can see the z-average diameter, which is the weighted average of your sample in the different buffers. You can see how that increases as aggregates are present in your sample. PDI, or polydispersity index, is also reported. In the commercial formulation buffer, the sample has a PDI less than 0.1, which is desirable and indicates a monodispersed protein. In the acidic buffer, the PDI is above 0.2, which indicates a highly polydispersed sample. This gives us qualitative information with the distributions at the top of the graph, as well as quantitative metrics with the z-average diameter and PDI to understand that reformulating this antibody from its commercial buffer to a citrate phosphate acidic buffer was a bad idea and is likely to cause aggregation. If we want to explore other buffers quantitatively, we can use what's called a B22KD assay. In this case, B22 is the second virial coefficient, and KD is the diffusion interaction parameter. In a B22KD study, a range of concentrations is loaded onto Stunner, and it will use UV-Vis to understand those concentrations directly. In this case, we're using 4 concentrations between about 1 mg per mil and about 10 mg per mil. We're using those across 2 different buffers right at the same time. So this is one plate with multiple replicates for each data point that you're seeing across 2 buffer conditions and 4 concentrations. But B22 and KD are measured at the same time in one assay. So we've measured concentration by UV-Vis. The other input comes from dynamic light scattering. When measuring these samples by dynamic light scattering, you can get size information, which is measured as a diffusion coefficient and leads to KD analysis. And you also get static light scattering information, which leads to this metric called Kc over R and has units of inverse molecular weight. So in this one experiment, you're measuring both axes in a KD-B22 experiment, and the purpose of it is to screen for protein self-self interactions in different formulations. What these graphs are showing us is the comparison of the self-self interactions in a succinate buffer versus a PBS buffer. What's desirable in a KD-B22 experiment is you want positive slopes and positive KD and B22 values. We're seeing that in our succinate buffer. This indicates that samples are having repulsive self-self interactions and are less likely to aggregate. On the other hand, PBS has a slightly negative KD, which means that self-self interactions are more neutral and aggregation is a higher risk. You can also have even worse performing buffers where you see strongly negative B22 and KD interactions, and aggregation is a great concern. One thing I also wanted to mention with this Stunner plate and analysis mode is that that type of analysis is compatible with automation. So as the world tends to move towards smaller volumes and higher throughputs for your analytical tools, the Stunner plate is compatible with plate loading and reading and everything that you need to do when you've integrated this with a liquid handler system. We also have an API software package available, which means that there's an ability to seamlessly integrate with other types of lab automation robotic systems. Okay, now let's pivot a bit. That was talking a lot about how you can use Stunner for characterization of biologics, whether that's looking at a central characterization lab, at-line analytics, or formulation studies. Now I want to consider the topic of antibody-drug conjugates. When I think about antibody-drug conjugates, a lot of the times the characterization of those is much more challenging when it comes to quantification than traditional antibodies that have not been conjugated. So that's kind of like watching an auto race where everything is a bit foggy and you can't see what's going on easily. To extend that analogy a little bit, classic antibody analysis is relatively straightforward when we're talking about quantification and sizing. But ADC analysis gets much harder. Thankfully, DLS for sizing is the same, but UV-Vis analysis, answering questions about how much do I have and what's my average drug to antibody ratio, can be a great challenge, especially when you're working with a novel molecule that you don't have a robust characterization package for yet. Again, Stunner comes to the rescue. Stunner has two different applications available to characterize ADCs for antibody concentration, drug concentration, and DAR by UV-Vis. The first one is a method that's been around for decades, and sometimes I'll call that old school DAR. In this technique, you need to know 4 different extinction coefficients for your molecules at 2 wavelengths. In other words, you have a drug extinction coefficient at 2 wavelengths, and you have an antibody extinction coefficient at the same 2 wavelengths. If you plug those known values into a system of equations, you can take absorbance spectra, like what's shown on the left here in gray, and understand how much absorbance is coming from your antibody, shown here in blue at 280 nanometers, how much absorbance is coming from your drug, shown in green, again at 280 nanometers, and how much absorbance is coming from your drug and antibody at another wavelength, where here we're quantifying at about 360 nanometers. If you know all of that, then the math becomes simple. Stunner will do it for you, and you can very easily understand concentrations of your antibody, your drug, and your average DAR. But that traditional UV-Vis method we felt needed more flexibility, since many times UV-Vis spectra in modern drug ADCs can look like this. When we have overlapping spectra, it can become a great challenge to characterize this, especially when you're dealing with a lot of samples that are novel or not well characterized yet. It can be a great challenge to understand which parts of the spectra are coming from your antibody and which parts of the spectra are coming from your drug or drug linker. So that overlapping spectra is a challenge because it's difficult to assign which part comes from which component of your ADC and how much comes from each component of your ADC. That means that when you don't know your extinction coefficients, which I'll represent with epsilon on these slides, that characterizing this molecule by UV-Vis can be impossible. So if unknown extinction coefficients are total deal breakers when it comes to UV-Vis characterization. How we decided to increase the flexibility of UV-Vis characterization is by giving users the ability, giving scientists the ability to decode your ADC spectrum. So here we're taking a commercial ADC, trastuzumab emtansine, that same spectrum you saw on the previous slide, And using our ADC decode application, you can break that signal down from the total absorbance shown in gray on the right into the correct absorbance coming from the antibody in blue and the absorbance coming from the drug in green. Once you're able to do that, quantifying antibody concentration becomes very simple, and drug concentrations can be understood as ODs or can be converted to concentrations and DARs if you know a single extinction coefficient, or you can run a single standard with a known DAR. Here's how that works out. If you take a known antibody spectra, you want to find its best fit within the ADC absorbance that you're measuring. So when you measure a gray ADC absorbance shown at the same, shown in both graphs on both sides of this slide, there's a lot of possibilities that we need to figure out a best fit for. So we have a maximum antibody spectrum, which is shown on the left, and a corresponding drug spectrum shown on the right. We know that this is our maximum because the blue antibody spectrum can't absorb more light than what we measured with the ADC. But in reality, the antibody spectra is likely lower than the maximum, and the complementary drug spectra higher. Here we're showing, as an example, 70% of the maximum antibody absorbance, and this is 50% of the maximum antibody absorbance. Notice the corresponding drug spectra climbing higher and higher, as expected. So to solve this problem of where the antibody spectra is the best fit, we've developed Stunner's ADC decode application to use absorbance features related to tryptophan in the 280 to 300 nanometer region to determine the optimal fit and the optimal antibody concentration. So Stunner can use this spectra directly in the ADC decode application, or you could store the resulting drug spectra through a simplified store spectrum experiment. Let's take a look at a couple of examples in a sort of hypothesized screening experiment where you may be looking at one antibody conjugated to 3 different drugs to create 3 different ADCs. In this experiment, we'll be evaluating 3 drug linkers, imtanzine, deruxtecan, and vedotin, and evaluating them by UV-Vis. The results of this experiment give us ADC measurements by UV-Vis that look like this. For deruxtecan, we do have a peak far away from that traditional antibody peak, but without more information on how that drug is absorbing around 280 nanometers, we're not able to figure out what the antibody concentration is. For the graphs on the left and the right, trastuzumab emtansine and vedotin, we have overlapping drug spectra, and analysis can be incredibly challenging. If we're screening our ADCs using our ADC decode application, it's able to understand the correct antibody concentration, figure out where that fit is in blue, and what the resulting drug spectra is in green. You can see that here. If we want to confirm this characterization approach is working correctly, we can do that with this method by comparing it against recorded spectra in the literature. So here we first evaluated trastuzumab emtansine and the same drug linker on a different antibody called narituximab, and compared the extracted emtansine spectra. You can see that their overlap, once normalized, looks incredibly good and in very good agreement. Next, we took that spectra and compared it to literature, which is now shown in gray, but again, it's very difficult to see because the gray and the green spectral lines overlap almost directly on top of each other. So this kind of approach does give you the power to understand what's in your ADC spectra in a couple of clicks, and it puts the power to do rapid, simple ADC characterization at your fingertips. This kind of analysis is ready for you as soon as Stunner is finished reading its samples, meaning that method development can be done incredibly rapidly, and you get answers to your conjugation experiments or screens incredibly fast. If we want to evaluate the performance of this further, we can look at those same three samples by the traditional two-wavelength method, where the extinction coefficients can be known, and by the ADC decode method, where we're evaluating the spectral deconvolution of the antibody when the drug spectrum is unknown. You can see that by both methods, there's excellent agreement in the drug and antibody concentrations, and we've run this across a dilution series where we're comparing 4 points of a 1 to 2 dilution series, and we see that the results are very, very linear and incredibly precise. If we take that same data and we want to evaluate how linear it is, because we're looking at so many samples, a convenient way to do that is to evaluate the ratio that drug concentration and antibody concentration are changing as we go across our 1 to 2 dilution series. Because we're diluting by a factor of 2x, we expect that drug concentration and antibody concentration would also change by 2x, and that's what we're seeing in the data. However, for DAR, that value actually should not change, so indeed across our dilution series, we get a consistent DAR value as we evaluate different dilution steps. And we want to evaluate our DAR results that we found versus the expected DAR results. And you can see those comparisons for the 3 antibodies for which we have a known DAR value given to us by the C of A on these molecules, and see that results agree within about 10% error. Stunner also comes prepared for situations when you don't know the molar extinction coefficient value of your drug. So in other words, you have a drug absorbance, but no way to calculate drug concentration. In this situation, Stunner will report the drug-antibody absorbance ratio, or DAAR, which is a ratio of the OD absorbance of your antibody and your drug, so that you still have a metric to compare different conditions or samples for your ADCs. To explain this a little bit further, on the slide we're looking at trastuzumab vedotin. In this case, we're able to easily calculate the molar extinction coefficient for the antibody trastuzumab at 280 nanometers. But we don't know the extinction coefficients for the drug at another wavelength. We don't know the extinction coefficient for the antibody at another wavelength. And we don't know the coefficient for the drug at 280 nanometers. So we kind of hit a hard wall when it comes to doing any kind of additional math. Because Stunner is able to understand the antibody concentration from the spectrum and the drug concentration from its ADC decode application, it's able to give a drug absorbance value, which is 3.12 OD, and it's also able to report what the optimal drug wavelength is that gives you the best signal to noise on that drug spectrum and the most signal to quantified drug. So in this case, we are looking at a drug antibody absorbance ratio of 0.4 that we can compare against other samples at different concentrations or different conjugation or process conditions. And we also know that when we do method development, that we should likely look at 250 nanometers relative to 280 nanometers to understand the different concentrations of the antibody and the drug in this sample. And if we want to correct this problem of not knowing the molar extinction coefficient values that we need to know, grabbing that data is easy using a known ADC sample. In this case, we've taken a trastuzumab and tanzine sample, run it through ADC decode, and we understand that for this sample, the OD at 252 nanometers is 5.87. Now, if we know the average DAR on this sample is 3.8 from other characterization techniques, it becomes a simple division problem to get to the molar extinction coefficient for this drug linker at 252 nanometers. So that means that our method development is done in about 2 sample runs. One to understand an average DAR on something like LCMS, and one on Stunner, where we can very quickly and easily understand the optimal wavelength to look at and the OD value that we have for our standard molecule. Stunner can also easily extend this type of analysis beyond antibody-drug conjugates to antibody-oligo conjugates, or really antibody-anything conjugates. For this type of analysis on ADC decode, all that's needed is a protein component of a conjugate and a non-protein component of a conjugate. So we can look at antibody dyes, antibody oligos, or indeed most other common conjugates that are out there. In the case of antibody oligos, you also have the advantage of having highly predictable absorbance from nucleic acids, so indeed the analysis becomes even easier. And as I discussed earlier during the antibody evaluation part of this talk, Stunner also comes with DLS reads simultaneous to a lot of its applications. So in this case, we've been talking about trastuzumab with 3 different drug linkers conjugated to it. I also have size information for those 3 same molecules read at the same time as my UV-Vis characterization reads to understand that my antibodies are intact or if aggregation is happening. So if we're looking at the data in front of us, which is mass distributions for those 3 molecules, I'm very happy with my trastuzumab emtansine and trastuzumab vedotin, but I can see that my trastuzumab deruxtecan has a wider distribution, a higher PDI, and might be well off if I reformulate it into a different buffer. So that's the kind of analysis that we've done before, looking at antibody drug or antibody dye conjugates to understand how samples can aggregate. In this case, we're looking at a sample where on day 1, we've evaluated DLS for sizing and seeing that intensity and mass distributions agree, that all of my sample is present at the correct size, the correct diameter for an antibody, or for an ADC, I should say. But after 7 days of storage at 4 degrees Celsius, I can see aggregation present in the sample at 1,000 nanometers diameter. Even though it's a low amount of mass, it is present there, and I need to be aware that this sample is one that can aggregate. So this kind of analysis is something that happens incredibly quickly on Stunner, as it's indeed the only plate-based system built for biologics to get UV-Vis and sizing information at the same time, incredibly rapidly. So here we're looking at in HER2, where we've gathered DAR information, sizing, and aggregation on 16 samples in only 10 minutes. And that's how Stunner can help you with an all-in-one biophysical readout on your antibody, biologic, and ADC samples, giving you sizing and aggregation information, protein quantification, drug quantification, and average DAR information. And finally, when you're keeping your protein and conjugate quality pit stops as quick as possible and as thorough as possible by keeping an eye on concentration, DAR, size, and aggregation on the same instrument done in only moments, then you're well set up to win the race to an accepted therapeutic. Thank you again for joining today's webinar, and I'm very happy to answer any questions that you may have.